I'm delighted to have with us today Rob Tanguay. He's going to give us. I'll keep my introduction to Rob very short. But before I go to the introduction, I want to remind all of you that this is part of the Nanotechnology Health Implications Research Consortium webinar series. And the main goal of this webinar series is to bring together not only the people that they try to assess the toxicological impact of engineered nanomaterials, which is the main focus of this consortium, but also to bring the application community to educate us on emerging nanomaterials, so we can really focus on safer by design formulations. So Rob is, again, I will keep the introductions short. He's a pioneer of the zebrafish model. He brought it to a level that we can use it for high throughput screening. He is well known in the field not only of nanotoxicology, but also in the broader chemical toxicology. And as I said, we're delighted to have him here. He's part of the NIR consortium as well. And his talk is the in vivo assessment of the nanobio interface to help develop safer nanomaterials. So Rob, I kept the introduction short as I promised, but you don't need an introduction.
Right, thanks, Bill. Okay, great. Okay, so first of all, thank you, Phil, for the invitation, and thank you all online and folks in the room for letting me speak today. So before I forget, I'd like to do big acknowledgments first. So the component of my lab, I have a huge lab. We have many different research directions, but these are the folks who contributed to the nano work that I'm going to talk about today. And then I have an engineering team, which really helps me build some of the instrumentation that we use to accelerate the way we do nanobio interactions and chemical bio interactions. And I have essential collaborators. I decided to choose a lot of this work as just to demonstrate potential of integrating material science, precision engineering materials with biology in a really powerful way. And then I have some really terrific collaborators at NC State, David Reif, and he really helps us with analytical work, analysis work in informatics, and he's just a really creative guy. And again, this is part of the Nanotechnology Health Implications Consortium, which is funded by NIH, but I'm also going to give some of the research from one of our NSF grants as well. Okay, so what are the opportunities when you think about why do we do what we do? So this idea of proactively developing chemicals, greener chemicals or greener nanomaterials isn't really new now. It was when we started working in this field. But the idea is really to proactively guide the development of safer materials for products. We certainly know our economy is based on high-performing products. Everything is based on it. So it's been my view, and by working with Jim Hutchinson at the University of Oregon, is I don't think toxicologists have been very good players in that role to help industry make better products. We all depend, all of our careers, all of our standard of living is dependent on this technology, yet toxicologists seem to want to shut down advancement. So I wanted to change the script. So we've been doing this for a long time. But the idea is not unlike from a chemical point of view and drug discovery. You really want to identify the physical chemical properties that drive the biological responses. It should be possible to do it. I'm not saying it's easy, but it should be possible to find the design rules that could predict biocompatibility or toxicity, depending on your perspective. So even though I'm actually not really a toxicologist, I'm a molecular biologist, I'm a biochemist by training, but I've been doing toxicology for a couple of decades now. But what I'm going to talk about today, I want you to think more about the nanoscience. So how can we use modern approaches in toxicology and systems toxicology to help nanoscientists? So we're not trying to find what is the mechanism by which one given nanomaterial produces an adverse effect. We can do that. That can keep me busy forever. But this is different. It's like, how can we enable a field to assess their innovations in a way so they can move forward? So the question is, how do you do that? And this is actually, all the innovation and the creativity that material scientists and chemists have, this is really exciting. They can't proceed in a proactive way if they don't have a biological platform or platforms or approaches to evaluate their materials. So we wanted to see if we could help. So the idea where we came in is we do this in the chemical space, but for nanomaterials, what would be the best system? So, all right, I'm going to pitch what I do because I'm going to say it's the best system, of course. But I chose the system because I think it's the best system. I'm not a born and raised zebrafish toxicologist. I'm like, I want to solve a problem. What are the best models to solve the problem? So if you're going to investigate whether a chemical or a nanomaterial has a unique property, or it could adversely or effectively modulate biology, the best time to do it, whether it's zebrafish, mice, humans, elephants, it doesn't matter. It would be during embryonic development. And there's a practical reason for that. We know that this is the most dynamic period of an organism's life. You're going from a single cell to cells migrating, interacting, differentiating, and gene expression. All the network that many of us study in different contexts is happening in early life. And it's all kind of on autopilot after fertilization. So literally all of life, all of biological processes are called upon during development. That means all biological targets are present at some point in development. So if you don't know what your target is, you have an unknown chemical, you have biology, which is kind of a black box. Let's join the two together. Throw the complex material onto the complex biology. It also happens to be the period of time from, if you look at comparative genomics and processes where the mechanisms between, say, all other vertebrates or even invertebrates and humans are most well conserved is during those early developmental events. And again, I mentioned everything is expressed at some point in time. So what, again, from a practical point of view, thinking using this system as a biosensor, using zebrafish as the ultimate biosensor, you should have the fewest possible blind spots to your test agent. Because the only way a test agent could impact biology is if it hits something. It hits a gene product and modulates it in some way. And then that gene product is integrated with biology, and that's when you can see an endpoint or a phenotype. So in a practical sense, if a chemical or a nanomaterial produces some effect, some change, it must influence an important pathway or process. Okay, so that's our goal from today's talk is going to be use this complex biology and look for a change, and then use the change to identify the target, and then also assist in the design of new products, if that makes sense. So why the zebra? There's some practical stuff. Some folks online have probably seen me give dozens of talks, but there are a lot of really inherent advantages. So we're looking at a 384 well plate. You're looking down at 4 wells. You have 4 whole vertebrates developing in front of you in 25 microliters of water, and they aren't even touching. So all of this, you got to remember, this is like vertebrates are developing in this tiny space, and the processes that are ongoing here are remarkably similar to what would be happening in a developing child. So you just have to remember that. And then so the scaling. So there's an advantages. Obviously, size is one. Imaging modalities are in your favor. For nanoscience investigations, for example, we talked a little bit earlier about creating materials in a precise way at scale is challenging. We don't have that challenge. We can investigate an entire animal with nanogram quantities of test material. So, but there are some considerations that we had to think about when we applied our ability to nano. Again, of course, the goal here is, in this case, is to investigate interactions and responses. So that means you want to get your material in contact with your biology. And if you believe me that the idea that all of biology is called upon during the course of development, from this one cell stage to full organ development, you don't know when your chemical or nanomaterial might be active, then hit it. You got to get in it as fast as you can. So you have to present it to this biological space early. So that's a design criteria. And again, you're forcing it. So this isn't meant to be an environmentally relevant situation. It's a way to again, a biosensor. So one thing that we discovered decades ago is that the presence of the chorion is a challenge, particularly for nanomaterials. So there's an acellular membrane that this embryo is developing, kind of in this protected little sac, and many of there are pore sizes in these membranes, but we certainly have demonstrated that this acellular membrane can be a barrier from nanomaterials. So you're not getting an interaction between this complex biology and your test agent because it can't touch it. So we had to come up with first methods to remove that chorion so we can force these interactions. And then we had to create instruments to do it for us because we needed to scale it up. We certainly know nanomaterials are different than, in many ways, than chemicals, is that the behavior of the particles change over time. So this center does a fantastic job of trying to characterize these transformations that occur in test solutions. And this is really stymied a lot of toxicologists for a long time. So then we thought, because we're not constrained by, which really fun being in the field kind of at the beginning, we're not constrained by what anyone else has done. So you can just do what you want to do. And so we thought maybe we can adjust the assay to tune it to the material, not the other way around. So we want to think, what can we do to our assay to make it more amenable to assess these really incredible materials that you guys are making? But then there's some kind of like conceptual ideas. When I think about how a material behaves in any environment, absolutely, and there's a lot of debate on this point, but I'm going to push it anyhow. It depends initially on its intrinsic properties. And I think that's the same for chemicals as well. And so that means if you're in this, buckyball as an example, the reason that these materials are going to aggregate, it's because of the intrinsic properties of this material, right? Of course it is. Whether if you have a metal oxide, whether it dissolves, it's dependent on the properties of the material. How it interacts with the environment is initially, at least initially, a result of the properties of the material. And of course, the environment it lands in influences interaction, but still, it's all driven initially by this structure, and the fate and partitioning as well. I'm only going to talk about this today. So how it interacts with biology is again intrinsically programmed in the structure, and then the fate, and then the biological response. And don't mean to make it sound simple that we can understand this relationship between these intrinsic properties and these interactions, but it should be possible to explain them. So the goal, of course, is to do that. So when Jim and I started working together like 12 years ago, really we came up with this concept, and could we put it into practice? So Jim will design a material platform, and many of my other collaborators do this as well. They synthesize it, do careful precision engineering of the material, and then the idea is you want to evaluate it. You want to get it in a system where you can evaluate the impact of your structure and your features. And then you want to give this information back to the designers, if you will, the material scientists or the chemists, quickly, so they can interpret, I made this, you saw this. All right, and then say, okay, can you redesign it, and then just close this loop and accelerate this design and reassessment in weeks rather than decades. And what could we learn by closing this loop faster? So this is what we wanted to do. And the idea in the greener nano is the idea similar to chemicals, but I think there's certainly more tunable properties of nanomaterials. When you think about the compositional changes you can make and the size changes and the functionalization, so there's a lot of, this is where all the fun is. If I was a material science, if I could do that, I think I'd find it very fun, but I'll let others do that for me. So how do we do this? So what we had to do, we've been testing chemicals for about 20 years now. So we have gained confidence that the data we get is valuable and translatable. And there's dozens and dozens of examples I could share at another time. So how could we actually accelerate it so we can impact more fields? So the idea is for a number of reasons, we came up with a timeline. So the typical experimental design, after we get our eggs, and what you're seeing in this picture below is some custom racks that we built to automate the raising of zebrafish in bulk. And then not only that, the ability to get the embryos, which is kind of our feedstock for our testing, in an automated fashion. So we can go from, these tanks, we can get 8,000 to 20,000 eggs in a minute. That used to take hours to do in the past, so we do this every single day. So anyhow, the timeline is we want to get these eggs, get them ready, and in this timeline, we clean them up in the laboratory, get rid of the bad eggs, and then we decorionate the embryos when they're 4 hours old. So we have an instrument that does that. We call it the decorionator. So we have to do this in mass, and then we initiate the exposures at 6 hours post-fertilization, whether it's nanomaterials or chemicals, and then we do a static non-renewal for most materials. We also have, and I explained this to somebody this morning, that we also have an aggressive mixing, which is very important for these types of materials that occur. And then that exposure is from 6 hours all the way to 5 days. And so that's what's happening in the dish. But what's happening in the embryo is you're going from this, a few cells on top of a large yolk ball to this animal that has a circulatory system functioning, a brain, a pancreas, a spleen, a kidney, and this happened in 5 days. So again, we have a pathogen-free facility. Once these embryos are dechlorinated I didn't show, then we have robots that can pick them up one at a time and put them into multi-well plates. And then various ways that we do the chemical exposures when we're done here. And then we have assays. I'm not going to get into the details. We don't have time today, where we look at the earliest behavior that you can assess in zebrafish. It's a Rohan beard cell-driven spontaneous motion, where basically we're looking at whether or not the exposure to our test agent affects the development of this behavior or the actual parameters of the behavior, either the background, the excitatory phase with this light pulse. It's not important. But it's a really sensitive indicator of whether development has been perturbed by your exposure. It doesn't tell you anything about the mechanism. That's not the goal today. The goal is to use the system to say whether the material can do anything by interacting with biology. And then we let development continue again for another 4 days. And at this point, these animals are pretty sophisticated. They can respond to a number of behavioral perturbations that we've automated as well. And then at the end of that, then we euthanize all the fish, and then we're doing these morphometric measures, and we collect all this data. All of this is fully automated, all barcoded, all managed by a LIMS system, and the reports are generated immediately. So what this does when you can automate it is I think I have it on the next slide, is you do everything differently when the you can scale up. So first of all, we can test broad concentration ranges. So maybe 6 to 11 different concentration ranges. Obviously, your negative, positive controls. We do a total of 32 replicate animals at every concentration. So that's massively overpowered compared to the mammalian counterpart. And we actually look for plate effects by doing this dilution series typically, and then we duplicate the plate. So we can now we have methods that we can randomize everything. And then we measure 22 individual endpoints. Every one is measured independently. So it allows you to track one animal and so you know the material, structure, the concentration, and you know what that did across this developmental lifespan for every material at every concentration. So the amount of data mining that you can do is this is where David Reif has been really helpful. So we can scale up. But what do we look for, right? This is where I think a lot of the angst happens when people say, why would you use zebrafish, right? Because so I'm going to spend a little bit of time on it. So we think about this as a really high content system. So a lot of the push is to move to in vitro platforms to realize the goals of the tox testing for the 21st century that many of you are aware of. But you lack complexity in cell in vitro system. We all recognize that. So what do we look for? We look at the impacts of thousands, if not millions, of interactions that need to occur to have a normal animal. So there are lots of ways to perturb this system. But when you perturb the system, you see a lot of common responses. And this is from experiences of screening thousands and thousands of chemicals across concentration. So you get these common responses, but these are initiated by specific hits. So you can take a pharmaceutical that has a unbelievably specific mode of action, and you do this during early development, and that target is integrated with biology, and you disrupted it. You've just broken the entire normal process of development. And what happens is the animal suffers the consequence. And the consequences, even though you have a very specific insult, ends up looking like this, very common. You'll see things like edemas of the yolk and the heart. The axis is bent in the animal. You get malformations of the fin and the eye diameter, circulation failure, heartbeat, blah, blah, blah. So you see all of these effects and changes in behavior. These don't tell you anything of what you hit, what target, or how it happened. All it tells you is take a closer look. And although it may be a drawback that you can't assign an endpoint, but from a screening point of view, from a biosensor point of view, it's a huge advantage. Now you can push thousands and thousands of chemicals through, knowing that you're not going to miss anything, because numerous modes of actions will lead to these effects. And in fact, some of these we statistically, and David has helped us with this, identified relationships between the endpoints. So we actually think that we call them super endpoints. we could probably assess 8 of these and not miss any bioactive molecules. That's a longer discussion. So, okay, so going back to this. So how can we test this? So Jim has a lot of expertise in synthesis. So we decided let's pick an architecture which will allow us to look at isolated features, size, surface functionality, completeness of functionality. And we picked gold for a number of reasons. Gold is certainly medical applications are obvious, but also if there is dissolution, the ions aren't toxic. We're not going to worry with silver. So there's a lot of reasons to pick gold, but it allowed us to tune this so he can modulate this with ultimate precision so we can know what's the influence if, and then again go through this paradigm. And then what can we learn from this? So I'm picking is probably what people would call first generation nanomaterials, the boring stuff, the boring spherical metal oxides. But I hope I convince you that there's a lot to learn in the material science space still by studying these materials and coupling it with biology. So in the first library that Jim went through, so we have a various size cores. We stay in the small space for a number of reasons, 0.8 to 10 nanometers cores, and then you functionalize at the R groups with a number of these different ligand shells and ask the influence in a very systematic way. One common biological system done exactly the same way. What's the influence of these changes? When you start thinking about you'll see in the literature, if you're a material scientist, you probably think about scaling surface area in atoms, but most toxicologists don't. So this is, how different are these? 0.8 to 10 nanometers. That's not that different. Well, actually, when you start thinking about what you're working with, 0.8 nanometers is only 11 gold atoms, and only 10 ligands can fit. Right? You just go to 1.5 nanometers. Now you're talking 35 ligands and 100 atoms. And then just by going up to 10, you're talking to 1,400 ligands. So what the biology sees is vastly different between these, even though compositionally they can be the same, right? That's probably going to be important to remember. So we screened a bunch of these. Some of this we published a long time ago, but I still think it illustrates a point. So we picked these 3. So I'm going to only talk about one core diameter for today's talk, but pick these various ligands that we're going to call either neutral, cationic, or anionic, and so we're MEEE or TMAT or MES. I'm going to use these in a couple of examples that I'm going to give you, and just evaluate them. Again, same core size. They're not dissolving. We know that, and then I'm going to show some of the characterization work we did. So we're really isolating what's the impact of that surface functionality. Can we use this platform? And this is one way of looking at the data. I'll try not to confuse you with all the other methods, but what you're looking at here is just a % of the animals that are responding in a group, and from 0 to 100%. And then you're looking at black bar means these embryos were killed by the exposure. Gray bar is some of those other endpoints that I mentioned, the edemas and the axis changes. That's what the gray bar. And unaffected would be the normal animals. And should be obvious. You look at this one, for example, the MEEE, even when we go to outrageously high concentrations, 250 mg per kg, nothing happens. These guys aren't crashing out of solution. These guys are just as stable in solution as these. So these guys, we're calling, these are benign. Now they become our negative control. And then we have these materials that as you increase the concentration from a low concentration to high concentration, you get an increased incidence of these adverse effects, but very little mortality. And then if you go to the positive controlled one, the TMAT ones, you increase the concentration, and then you get these effects, and then it goes to mortality. So again, cores is exactly the same, composition exactly the same of the core, the shell's the same. The only thing that's different is the functionality. And we can already use this biological system to say these guys are behaving differently. So that was the one finding that and we published a number of papers on this. Some of the specific effects of TMAT we published, but it just gave us confidence that this is going to be a valuable approach to use the zebrafish solely to help Jim design different materials. But then we started having this issue of dispersion, which many people do. So a lot of folks who do nano-bio interactions with materials that they can't get in solution, the most common approach is mix it with something that coats it to improve the dispersion. I really don't like that approach, for obvious reasons, right? So unless you have a dissolving material, what's interacting between your biological system is the surface. So if you have an interesting material, you want to know its biocompatibility, and now you code it with PEG. Okay, that's really, and that doesn't mean you know that the PEG is going to stay present and you're not going to get that exchange in the system, but it's really a bad idea, in my opinion, to try to assess surface effects when you've changed the surface, even though it's not covalent. But so anyhow, can we get around that? So we thought about that. So we were pushing through these libraries, and suddenly we had some that were not monodispersed. They weren't stable. And I'm like, okay, does that mean we just can't test it? Maybe so. So what we and others certainly have realized over the years is that, you know, the way these materials behave, their macroscopic properties behave, really depends on the conditions of the media. And certainly for many of them, aggregation, depending on the charge, often occurs at high ionic strength. So people who are doing cell-based studies, cell culture studies with DMEM, et cetera, you get a lot of this aggregation and crash out. And then, of course, the biological response is altered. If you have materials that, when we talked to Phil last night, materials that float, right, then you don't have contact, that's going to interpret your result. You have materials that aggregate and crash out, there's no exposure. So how do you interpret your results if you're not paying attention to what's happening with your materials? So it's really necessary to characterize what's happening to your material in your exposure conditions, and don't use your pristine characterization data to make your assumptions. So what we decided, this particular material, which I'm going to call 1.5 nanometer, it's a gold mercaptopropionic functional group. This thing crashes out of solution very rapidly, and you can see this in a simple UV spectra. So if you look at time 0, and you can see a really nice absorption across wavelengths. But and there's a reason we picked these 2 time points. Our assay is 114 hours long. So within 18 hours, and actually much faster than that, this stuff is crashed out. It's at the bottom of the dish. So I can't interpret the results when these materials are just clustered in these little marbles on the bottom. So clearly what's happening? These characterizations, like many that I get from Phil, are done in water, right? So we have great characterization data in water, but how many biological systems are tested in just water? So we asked the crazy question, well, when do they crash out? So with this example, what we did is our embryo media is, it's not salt water, it's more similar to media in terms of its conductivity. So 100% embryo media, you can see within 18 hours at both concentrations, a lower and the higher concentration, 95% of it has already crashed out of solution. So we said, well, let's just dilute it out and then measure how much stays in solution over time. And then this is our standard condition. This is why it's crashing out. But if you start diluting it, by factors of 5, you can see that when you get down to just 20% embryo media, it's much more stable. So when you get down to just water, it's stable for 5 days. So we can keep these monodispersed if you could control the ion content. How do you do that when a biological system depends on ions? So then I asked my student at the time, Lisa Trong, can zebrafish tolerate living in water? Can we adapt our assay for the material scientists? And one would think there's no way that these guys can develop in just water. So the idea is we tried it. So first what we did is this is, again, our exposure paradigm that I showed you. So this is where development is trying to happen in vertebrates. And then we just diluted the embryo media just like we did here, but now do it in the presence of zebrafish. And then measure one embryo in a well, do multiple replications, and then look for these high throughput evaluations that I described, including behavior. So look for anything that might be different because you just diluted the salt on these guys. And what we found, surprisingly, so this is our normal condition. There's always a little bit of background mortality. Animals are normal. That's what our assay tells you. And you start diluting our media with just DI water all the way to 0. These guys are fine. These guys, I'm not saying these fish are normal after this. There are problems. But within this, accomplishing all the complexes tasks of biology, they can do it in just DI water, which was really important. So, and this is kind of what they look like to prove that they look like their friends. But why is this important, right? So if we would have just, and there are some labs that do this, just throw your materials in a dish and then get a response and say, this is the concentration response curve, and not really pay attention to what the material is doing while you're doing that. So in 100% EM, we would interpret these materials as not toxic, not biologically active. They're pretty much normal, a little bit of background, but it's pretty much normal. As you start diluting the ions, the exposure concentration is exactly the same, dilute the ions, and you start seeing these profiles change. Suddenly, when you get down to this concentration, look at all the animals that are actually dying or getting malformed. And you keep going, and that concentration response changes. When you're back to DI water, this material, which we would have deemed completely safe, is actually one of the more toxic materials we've ever tested when you make it bioavailable. So this is a cautionary tale, and I think more people need to pay attention to. We also did obvious controls. We took another material that wasn't toxic and make sure it didn't become toxic, and we put it in water, and it's not. So it's clearly the material that's changing, not the water. And we've tested now hundreds, if not thousands, of different nanomaterials, and in DI water we can do this. This kind of just shows you what do they look like. So if you touch these guys, this is done when they're done in water. This is what you touch them on the tail, they move around. If you touch this guy in the tail, it just can't move. So development happened, but this behavior has been affected specifically from this nanomaterial exposure. And this biological activity that we detect here was completely masked by the aggregation of the material. So I think we're in a better position to assess these. Okay, another case example. Again, a boring gold particle, but a really interesting finding. So we wanted to explore the influence of subtle surface variation. So if you have all those ligands, you can do ligand exchange. If you don't allow it to go to completion in a very, very controlled way, what's the biological impact? So this paper just came out recently. So the idea, again, is back to this gold platform, and with the two different functional groups, this MES and then the TMAT, and then we can have the idea that most of it's a ligand exchange with phosphine here. So you exchange the ligand. So when it's fully ligand exchanged, there will be no phosphine groups when it's complete. And then you can, Jim is able to make these where you can very carefully control the degree at which the ligand exchange was not complete, therefore leaving these defects of phosphine on the group. And just ask the question, what's the impact biologically? And I don't have a lot of time to show you all the characterization. It doesn't change the size. It doesn't change the stability in solution. The only thing that's changed is the surface. And we can do this if you can precisely control the chemistry, and he did. This kind of shows you the size. So you guys can really make these really tight. Distributions are tight. Sizes are confirmed. And then the functionality, the other characterization methods for looking at the ligands. But so if you really think that there might be an influence of surface defects, you need to be able to synthesize them with a precise degree of defects and then characterize them. So what we did is, this is a hypothetical, so you can have this TMAT or the MES ligands, and then vary the degree of full ligand exchange and some where it's not complete. So normally you'd like this to be 100%, and then this would be 0. But if you can program in your synthetic methods more defects, then this percentage will go up. And then you have to prove that you can make it and characterize it. So using a proton NMR, you can actually measure the degree when this increased percentage, you can start seeing this the phosphorus group pick up. You can see this small amount picking up. So you actually can detect it, and it's a small percentage. And you're not doing single particle analysis. You're doing it in a pool. But we can make it so within our pool, in this example, 74% is fully exchanged, and there's about a 25% defect on it. And then what's the impact biologically? And what we did is there's huge differences. And we didn't expect this. So if you did, the top one is just the kind of traveling through the phosphine space. So if you go to the MES where it's fully functionalized, we have no toxicity. You're looking at a summary plot that shows all the individual endpoints. And basically all your eye needs to look at, whenever there's anything red, that's statistically significant. And this is a particular endpoint here is mortality. So as you increase the phosphine percentage, the toxicity goes up. That occurs whether it's an MES or if it's a TMAT. So it's the defect itself that is driving the change in the biological activity. So there are some practical considerations there. So if you're going to make a material, and this is going to apply to other materials, you need to get your functionalization to completion. You can't have this mixed bag. And if you don't thoroughly characterize your material, you may have this going on. So you may interpret a toxic result from your material, and it may be from some minor constituent in your population, which is how we discovered it in the first place. So these very subtle variations can really cause an effect. We published a paper a few years ago demonstrating for this TMAT response, these gold particles, that you get this really intense cell death in the eye. This is actually a transgenic animal that we got from Dr. Randy Peterson, where you're looking at in vivo apoptotic activation, and we see it in the eye. And you don't have time to look at it, but you can see it's the higher degree of the phosphine group, the more intense the cell death is. So that's mechanistically, it's inducing cell death, and that applies to the TMAT and the MES. So that's if you also look at some oxidative stress response genes, which we do routinely, you increase the degree. This is in the MES, and this is a TMAT example. As you increase the amount of incomplete ligand exchange in phosphine, you get an increase in oxidative stress. So it seems like these are just more reactive in causing oxidative stress, and this is kind of a closer look at the genes. So that seems to be the mechanism. But again, this is from a material science point of view. If you can synthesize these variations, then you can evaluate them and then understand biologically why it matters. Next one is another one we stumbled upon with these boring gold particles that nobody cares about anymore. So a few years ago, maybe 4 years ago, we changed our workflow in our high throughput screening facility to, instead of doing liquid handlers to do our liquid exchange, we went to this bioprinter. So the pharmaceutical industry uses this. So it's an inkjet technology printer from HP. And what you're doing is, you see that blue little well? It's a small area, like it's where ink would be injected at your home printer. But you put your concentrated stock of solution, typically in dimethyl sulfoxide for chemicals. And then that DMSO breaks the surface tension of the water and gets it to the thermal injection point, and it dispenses 13 picoliter droplets of your chemical. And then it counts the number of drops absolutely precisely to deliver the dose. The dosimetry in our system is phenomenal because of this. Okay, so we never applied this to Nano. So we were working with this new materials with Jim, and I said, well, let's just run some of our old materials through. And saw a very different response. And we're like, what's going on? So we go back. So now we implemented this years ago for chemistry, and we tried it with the Nano, and now we're cautious to do this for any other material. And this is why. So going back to that material, this MEEE, this was our benign one that we can throw 250 mg per kg at the system, and we never see anything. And this is a good example of it. You're looking at a mortality within 24 hours from low to high concentration. Nothing's happening. You do that exact dosimetry in the bioprinter, and these animals get killed. Okay, so we know it's not the bioprinter itself killing it. So it's like, what's different about this exposure and this exposure? It's not the biology. And what we figured out, of course, is that when we were working with HP developing this aqueous handling, because we developed some protocols for them for real-time PCR. So this device was meant to print inks, which are in solvents, low surface tension, not a problem. You try to put water in this with these small chambers, you guys know what happens there. The surface tension allows a little barrier so you don't get your chemical to the bottom. So we worked with them. They came up with surfactant solution. So now you have to put a small percentage of surfactant into your test solution to break the surface tension in order to deposit your chemicals. So they use tween 20. So our hypothesis was maybe it's the tween 20 interacting with the material, and then that's what's changing the material and changing its properties. So again, they picked tween 20 for a good reason, right? Because they're mostly driving for the pharma industry. So it's a food grade additive. It's safe at these concentrations. When we actually measured the impact of adding tween in terms of these macro measurements of absorption and DLS, et cetera, we don't see any change. But this stuff is just in the back of your mind. These are in commercial products all the time. So you're going to be interacting your nanomaterial that you're putting in your product with these surfactants in the actual world applications. They're not pristine. Just put that in the back of your mind. So ice cream, not that I wanted to know there was tween in my ice cream, but there is. So there is tween. So it's a co-exposure. You have your water, you have your nanomaterial, and a very trace amount of tween 20 in this reaction, and then that's being deposited in your cells. So what we found was, this is what the functional group looks like, and we tested, and the paper describes this. We tested a number of other surfactants, and we see the same thing. And so this is kind of looking at synergistic effects. So this is if you look at the concentration of tween and the nanomaterials with tween is in the dark circle. So if you increase the concentration of just tween, if you go a high enough concentration, you might imagine surfactants will dissolve an embryo with a high enough concentration. So when you get a high concentration of tween on itself, it'll cause toxicity. But what you see is a synergism between the nanomaterial, which was never toxic before. We've done this literally hundreds, if not thousands of times. And suddenly, in the presence of tween, as you increase the tween concentration, it becomes toxic. So the question was, how is this happening? And just some of the obvious things might come to mind. Maybe it's assisting delivery into the cells. We looked at that. That's not the case. But is the tween, that low concentration tween, changing the properties of the materials in solution? That was the predominant hypothesis. And I don't have time to show all of it, but in fact, it did. So what happens? So so we know that there's a kind of a tween corona that goes around these, but in fact, what happens is these sub-aggregations. So Aurora Ginsberg and Jim's group used diffusion ordered spectroscopy DOSY NMR to monitor the size change, the diameter change of these particles, plus and minus tween in a dynamic way. So we absolutely know the size changes of these aggregates. They're very small, and they're too small to do it in DLS, but because they're very tiny. So we can, with pretty good precision, we know that what's happening. These things are growing, and we also know the biology is saying they're growing and they're more toxic. We also measured differences in uptake. We certainly suspected that maybe the tween would affect its uptake, and it does slightly. So this just shows the amount of gold particles with INAA analysis, and then you get a modest increase of the amount of gold that comes up with the tween. That this didn't explain it. It really seems like it's actually the material surfaces change, and now they're more reactive, substantially more reactive. From a material that we could never make toxic. We've done this in cell culture cells. We've done it in zebrafish, and now we can create a toxic material. And again, why it's important is you don't have to test in formulations, right? There are a lot of emulsifiers. There's a lot of surfactants in consumer products. And the nanomaterials, say sunscreens, the easy example. Sunscreens are effective, but if there's interactions with some of those other non-active components, that maybe it's changing the properties of your creams or whatever, it actually could be modulating the properties of your materials. And those are the ones that are going to be in contact in the environment or the consumer. And this is a challenge, and I think we're going to have to think about, we're not going to be able to test them all. That's down that same path. We don't want to go now create a testing paradigm. Instead, we have to predict it. So can we get to the point where if you know surfactants are in your mixture and you know enough about your material, what might the interactions be? And start thinking about proactively, even at that level of designing your materials. So I think this boring gold material that no one wants to talk about anymore is really pointing out when you can control the precision synthesis in such a way that you can isolate and look at these properties, you can understand them, and then you can use them to design better materials. So last one I talk about is part of the consortium. So Phil's group is providing and characterizing a phenomenal group of materials. And our goal as the other investigators are using our expertise and our systems to evaluate those same materials in different systems. And hopefully stakeholders like public health issues and regulatory needs will be addressed when we can start predicting this. That's the overall goal. So obviously what I'm doing is I'm evaluating these in the zebrafish model. So we've implemented the method that we're described in terms of uniformly dispersing these materials, and we've done this for all of the materials so far that we've received. We've also compared it with the data that provided by this center, by all the synthesis methods that were provided in our data sheets. We've confirmed the DLS and the zeta potential, and they're really spot on. So they're doing a good job. We can recreate it. So that's great. And then this kind of, if you start looking at the first group of 25 engineered nanomaterials, these are the various categories and the number of the materials. So we've tested most of these. There's some that we haven't. And so I'm not going to plow through a bunch of negative data, but I want to show you, again, the way we did it was similarly. We decided a concentration range for these materials between 0 and 50 micrograms per mil, and we don't go past 50 we never will. when we have more information about surface area, we can start correcting these doses based on those properties. And again, we do N of 32, and again, we do ultra pure water for every one of these unless they're in something else and we get them. And again, we look at these assays in 5 days. This is a way, kind of a heat dot plot summary. So you're looking at, these are all those endpoints that I described, and these are the example materials and their names here. And then what you're looking at, anytime where you see white, that doesn't mean we didn't do it. We evaluated thousands and thousands of animals, and we see there's no adverse effects at all. We measured a couple different behavioral assessments, and we do see a smattering of activity for some of these materials at higher concentrations. We don't know exactly how to move forward and to trigger whether these are a concern. I'll show you what we're actually doing. But there are only a couple materials that seem to act like biologically active materials. These are the silver silica, either 4 or 16%. And you can see that there's a lot of different endpoints are affected by these materials. So this is very typical that we see for something that has some biological activity. And there's really not much going on here. So most of these we're putting to bed and we're not doing anything else. And then we're focusing on the 2D materials that are coming next. But what's really nice is that we can do these whole animal, most sensitive system that we can imagine across concentration, massive replication in just a few days, which is powerful. So then but just to tease you a little bit of what we're actually doing. So what if we do these assessments, we do these early lifetime exposures, we don't see anything, or we see something at high concentrations, but maybe we're missing something. Maybe we're missing an interaction that's important for public health. So what we decided to do to kind of guard against that is this paradigm. Can we do, one example, we can do our engineered nanomaterial exposures again from this window, and then take away the chemical, take away the nanomaterial, and just raise them in mass until they're about 90 days of age. So we go into 90 days because at this point they're sexually mature, so they're done, and now they're just going to get old like us. And then ask whether or not, again, the only exposures, you have complete control when to start the exposures and for the most part, when to stop them. So you might do this entire 5 days, you might do for 1 hour, you might do for 2 hours, so you can manipulate this. By adjusting the exposure window, you're actually probing different aspects of biology. So anyhow, with this example, it's all of development. And then just raise them up and see if you see anything. So we have absolutely no hypothesis in advance that any of these materials might interact with any target to produce any of these long-term effects. Well, let's take a look. And so we've done this for lots of chemicals, and now we've done it for about 9 of the nanomaterials. So what do we look for? So I brought these up because many people don't believe you can do these sophisticated, relevant measures in fish. But so we've automated the ability to look at impacts on fitness, and we do this using this one's commercially available. It's you're measuring oxygen consumption, and you can calculate BMI, et cetera, when these guys are exercising. So we do that pretty quickly now. You can measure anxiety and or fear, aggression, social interactions, and actually learning. So these are words intentionally selected to make you think about stuff that we're worried about in, say, kids. So again, we use that same paradigm, and then we have to do many of these exposures, had to figure out how to do that, and then how to rinse them effectively, and then how to raise them, and then the record keeping is just a nightmare. Because if you do this in rodents, you have this ability to do little chips, right? You can chip them and scan them. You really can't do that in fish. So this ends up taking a massive facility to do, and we have that. This is just one example of fitness. So this is just a treadmill for fish. So you're looking at the belt is being driven by this propeller on this end. So you can change the speed and change the current. And what you're looking at is kind of at a medium speed. These guys and girls are taking turns being the lead car in the race. And they actually draft behind each other. It's kind of funny watching them. But as they're exercising, when we're telling them to run faster, this is a really sensitive oxygen probe here. So you're measuring oxygen consumption by the entire group. And then you could purge it, to reoxygenate the water, and then change the speed. And then you see how much harder they have to work. So we've done this for a number of chemicals, and we've identified some chemicals that, from a developmental exposure, they have to work substantially harder to achieve the same exercise as the unexposed. This down below is actually a feeding study. It's not a nanomaterial study, but this is actually hear that commercial on the radio for super beets, have these nitrates to increase cardiovascular. We created a diet where we're testing, and it works in fish too. So anyhow, so we're asking whether or not that transient developmental exposure affects fitness. Now again, it could be effects on energetics, right? Mitochondrial function, mitochondrial number, muscle, et cetera, or heart output. So that's one assay. This one's one of the most robust ones. So it's a anxiety test. So you can have these fish swimming around. This is an instrument that we custom developed. That's a camera looking down on these animals, and there's one animal per well. And this here is actually this surface is actually a TV screen, which is important in the next assay. And this surface is another TV screen, so they can actually look at the screen and interact with it. And the camera, the only thing the camera can do is what did the animal do in response to what we did to them. So in this case, we're stressing them out. So we have solenoids on the bottom of this that taps the tank, and then just like that. And what happens is they hear it from their ears, and they also hear it with the lateral line, and it's a startle response. So they, just like rodents and people, do the same thing. And then so you get this activity, you tap it, they get stressed out, and then they calm down. You tap them again, and then they habituate pretty quickly. A control animal will habituate after 3 or 4 taps, and they're like, I'm not wasting my energy because nothing's happening. So again, we're asking whether chemicals, this is really sensitive. We see chemicals and drug exposures that completely eliminate habituation. Again, this is exposure that happen when the CNS is developing, and then this is an effect that goes on through the lifetime. And in fact, for one of the PHs that we've studied, we just published this recently, that actually is an epigenetically transferred trait. So not only do you affect anxiety from a developmental only exposure, it's passed on to multiple generations. So it's a powerful assay. This is a fear response. So this may be brutal, but so again, these guys are looking at this video, and this is what they see. They're looking up and they're like, oh, what is that, right? So if you saw that and you can't get out of your tank, you have a choice. You can ignore it, try to interact with it, or hide in the corner. And what you can see here is you're looking at their likelihood to be near the video, so it's kind of random. You turn that video on, they see that image, and they get in the corner and they hide, right? And this is it's a biologically programmed response. And we don't know whether the material might interfere with that response, but we're looking. And if we do see it, then there's 2 things we can do. We can attack it from the biological side. What are the regions of the brain that drive those responses, so that the neuroscientists can help us there? But we're gonna dial back and know that, we know that it's during exposure when it happened. So we're gonna try to figure out during the exposure paradigm, how did the developing nervous system get hit to produce this effect? So it's a powerful system. And then this one is really looking at works here. Yeah, click that one. So this is another one, that same instrument, and they're looking at a schooling fish. So they'll put 4 fish, and they're swimming back and forth, and these fish are very social, and they actually interact with the screen. So they tend to be near the screen. So if a chemical makes an antisocial behavior, which is what we see in kids, we're worried about this, but we don't know what chemicals are providing it, we might be able to do that now. And then this is just another way, another instrument that's actually putting multiple fish in a tank. And what's really nice about this is it's measuring the motion of all of your animals in the tank, and it's also calculating the vector distance between each fish constantly. So whether they tend to want to be in a group or away from their group, you can automatically check it. And then so again, we don't know that it's going to affect us, but we can look. And this is the final one. We developed this instrument that it's assessing learning. So when you do learning tests in rodents and actually in people, or if you're a manager with your students, is you can be a manager with a stick or with a carrot, right? And so it's really hard to automate carrots, but it's really easy to automate sticks. So punishment. So we basically want the animal to associate a color with safety or not safety. So what they do is they can swim under this divider, and they can make a choice to be on this side or this side, and we can change the illumination color. And then when they make the wrong, and it's a beam crossers that detect this, it's not a camera. When the animal makes the right decision, they're not shocked. If they make the wrong decision, they are shocked. And so the system automatically, how long does it take them to figure that out? What's the period of time of shock? All this data is automatically collected. We do 30 to 50 trials in an automated fashion. and then we can get learning curves, and then we can ask whether the chemical, or in this case, the nanomaterial, is affected. So this is we have a whole room full of these now and more coming. So this is really a sensitive way to see whether or not can we identify unrecognized interactions between nanomaterial feature and impacts on the central nervous system. So with that, that's kind of a nutshell of what we're doing and how we're trying to use high throughput biology, kind of state-of-the-art technology to really help material scientists make better products. All right, thank you.
Presentation. Few questions. We start with the audience first, and then we'll switch to the people online. So any questions from the audience?
Thank you. Very interesting talk. I have one question. You talked about several generations of effects. Have you tested the effect of nanomaterials on second and third generation of the fish?
We have the exposed generation effects, including some of these behaviors. I didn't show you the data, but no, we have not, and we should, because we're really well positioned to do that. And in fact, if there was a material that there's a reason to suspect it more than another, in terms of the property, the composition, or then that's where we'll start. But it wasn't budgeted in our proposal to do that, but I have a couple students who are really interested in that, so maybe we should do that. Yeah.
Thank you.
I mean, this is very impressive, and I think from the screening point of view, from hazard identification and characterization, it's a great approach. How can we translate this for regulatory toxicology, though? I mean, how do we extrapolate from exposures to humans? Exposures to
it's a great question. I think that's the place to start first, and I think toxicology is getting a little bit better at that. If we could anchor any of our toxicological assessments, hazard identification, to some realistic exposure scenario, I think that's what we should do. All right, so that applies to this system, applies to mice, and so that's, but in terms of the findings, so say you got the dose metrics in a region that matters, and you get an effect here. We're not going to regulate, shouldn't regulate based on, a hyperactive response in zebrafish. No, it's the opposite. And instead, use these systems to find the materials that can do something, and then use the stuff I didn't talk about today, use genetics and other to identify the how. And then you can translate that into other systems, because I'm telling you, the mechanisms are going to be conserved. We have so much experience to demonstrate that. So don't try to regulate based on an endpoint. That's what Tox 21 is trying not to do, right? It's the pathway, the AOP concept. So it's really understand the mechanisms and then develop ways to translate those mechanistic questions into your systems that you are regulating on. That's how I would proceed. Yeah.
Thank you.
Okay. All right.